So in this lecture, we want to use a motivating example to sort of kind of explain what some of our big goals are for this module. So like Tabachnik and Fidel, we want to be short on theory and long on sort of understanding. And so what we're trying to kind of ultimately get to is sort of an understanding about what common techniques there are for multivariate data. What are the questions that they're useful for? How do you use them? So some practical like implementation, and then interpretation of their outputs, which we'll find is often much more challenging than when we deal with what we think of for fairly straightforward analyses that we've done in the past.
Yeah, so multivariate statistics can mean different things.
Depends who you ask.
It does depend who you ask about sort of whether all of these things categorize or whether it's more specific to one particular type. We are going to take the broad definition here because we think that's just fine. And so it's the multi-part. So any statistical model that includes multiple things, that could be multiple predictors. You will recognize this as something you've already seen before in the form of multiple regression. So sometimes people include that when they talk about multivariate statistics, although it's probably good for you to know that some people don't consider that.
Yeah, Tabachnik and Fidel do, so
yes.
That's why we do.
Yes. It can be no outcome variable, so you can just be exploring the relationships between a set of variables without trying to predict one, And it can also be multiple outcome variables. These two are the more common ones that will be referred to as multivariate statistics, where you have either nothing on the y side or many things on the y side.
And you could have just one thing on the x side in the last case there.
That's right.
So it's really just any combination of any sets of variables, really, where that set might just be the set with itself. And so you can think of this as now the even bigger generalization of all the things that we did in quantitative methods 1, where we were generally dealing with just one outcome variable and one or more predictors in most cases.
Yeah, and so the challenges to dealing with multivariate data, and particularly highly multivariate data, when you have many, many variables to be thinking about, is visualization is going to quickly become an issue. Remember, we as humans are limited to being able to picture things in 3 dimensions, and so once you go beyond that, things get hard. Some of the modeling can be challenging, and there's different things that you need to consider, which you will see much more of throughout this module. And then interpretation does tend to get a little bit more complex than when you're just thinking about how does this one thing relate to this one other thing.
Yeah, so you're sort of saying how does this set of things relate to this one thing or this set of other things?
Yeah, it requires you to start to understand this idea of shared variance and how that's influencing your results and all sorts of more complex things.
And we're going to talk about variance and covariance coming up.
Yes.
In a couple of lectures.
Okay, so let's think about an actual example, and one where, in particular, using multivariate statistics helps you to understand something biological that it would be that we don't have just a good straight-off measurement of on its own, and that is we're going to think about pitcher plants and pitcher plant morphology specifically. So this is a pitcher plant. If you haven't seen one before, these are carnivorous plants. They look like little pitchers, and insects end up in there and then sometimes get caught in the little digestive juices that are at the bottom of this pitcher, and then the plant gets a little bit of extra nutrients that way.
So the data come from a paper by Aaron Ellison and Elizabeth Farnsworth, where they were basically trying to relate sort of morphology of the pitchers to the soil and kind of other community around them, trying to figure out when plants prefer carnivory to other photosynthesis. We're just going to focus on just the morphology of the plants in this example.
Yeah. And so you can see we can measure all sorts of things about this pitcher plant, all sorts of individual little measurements in the goal to understand something a bit more holistic about the plant's morphology. And that's where multivariate statistics is really going to help us.
Right, so rather than measuring just one thing and trying to relate that to one other thing and doing that kind of in sequence, right, we're able to actually measure a bunch of things that might all have some correlation to each other, right? So you can imagine bigger plants are going to have a bigger circumference, but they're also going to have a bigger diameter and a bigger height, right? So there's all these things that if you were to just look at them individually, they might kind of all be telling you the same answer, but
each one might not totally encapsulate what you're trying to
what we're really interested in is what's the shape of this pitcher? So there's a lot of data in these in this study, and looking at effectively just a wall of data is not that helpful. Even looking at in sort of tabular form is not that helpful. But the sort of thing to sort of think about right now is how are the data organized?
Yeah, and to sort of note that, for a single individual, right, these measurements are, of course, not independent, right? These are different characteristics of this same individual. And so one of the goals of multivariate statistics is to try to understand those relationships.
Exactly. So we can visualize 3 of these and maybe add a 4th one. So we can look at mouth diameter, keel diameter, and tube diameter. And then we're going to color the points by plant height. And so you can see there looks like there's some relationship as, that they're sort of kind of all changing in the same direction, but they might be sort of related to how big the plant is overall. So here's sort of short plants in the dark colors, large, tall plants in the lighter colors. But even there, it's very challenging because we're limited now to just like 4 variables.
So let's try another technique. You've learned about ggSCATMAP before to look at pairwise relationships. And so we can use it to make a bunch of pairwise scatter plots and univariate density plots along the diagonal. And it'll also tell us about those pairwise correlations. And so that might get us a little bit further in trying to understand how all of these many variables relate to one another beyond just the 3 we plotted. So let's try it.
Very quickly we see that with 12 variables to look at,
it's a lot.
It's a
still,
lot, yeah.
Right?
So we're going to have to pick a subset of those, right? So even now, right, we're still going to have to sort of look at restrictive sets, and you might have to make multiple plots like this to really get a sense for the relationships between things.
Yeah, and one of the things that you'll find when you're dealing with multivariate data is one technique is to break it down like we're about to do so that you can understand parts of it. But then we'll also start thinking about things like Kevin said, variances and covariances and things like that are coming up. And those are ways for us to summarize the relationships among these variables when we get to a point like this where there's too much for us to use what you might be used to when you're just thinking about a couple things at a time.
Yeah, so let's just pick 4 to work with for our example. So height, mouth diameter, tube diameter, and keel diameter. And so these are the univariate distributions of those, and they look roughly normal. That's good. These are pairwise scatter plots, right? You can see that there's some relationships there. And then this upper triangle is pairwise correlations. And so you can see, like, for example, height and keel diameter have effectively no correlation, but height and mouth diameter have pretty high correlation, 0.6. And so this is a very quick way to kind of get a first feeling for how these variables might be related to each other, or in this case, unrelated to each other. But remember, it's not just that. We actually have 4 different sites, and this is what the original question in the paper was asking, was do these 4 sites have different morphologies, and are those morphologies correlated in some way with what's going on in the soil environment around the plant? And so we have 4 different sites, and so there's actually, for every one of these, there's 4 different correlations and 4 different densities. And so again, there might be different relationships between these different sites, and that's a question that we might want to investigate in a multivariate analysis.
Yeah, so what we're going to do now is we're going to think about different questions that we could address with this data set and different approaches that we could take with this data set. And we're still in the introductory part of this module, right? And so we're just going to introduce these here, which I just want to warn you that most of these we're going to go through in much more detail. So if you find yourself wanting to learn more or sort of being unsatisfied by the kind of brief introduction that you're about to get to these different approaches, know that those are going to be dealt with in more detail.
That's the next 4 units.
Yes. Okay. So first is just fitting a model with multiple predictors. So here we're going to be asking how some variable is able to predict an outcome variable. And this is specifically going to be after accounting for all other predictors. So remember what you learned about in multiple regression, that when we're looking at, okay, how does mouth diameter impact plant height, for example? Height is our focus in this case. That's going to be how does mouth diameter predict that after accounting for tube diameter, keel diameter, and site, right? Remember that that's how multiple regression works. That's how it's set up. It's doing as we've asked it to do. And that can be important for multivariate stats. more to come.
Yeah, and this is, we were worried about things like multicollinearity, and those were the sort of concerns that we had when we have multiple predictors. And so our correlations in the previous plot were too high, so
maybe it's all right.
We're probably okay here. And so if we run that and look at the output, we can see that mouth diameter is a great predictor of height, and pretty much nothing else is. And so we learned something. If we want to predict height for these pitcher plants, we might only need to measure. For this set of variables? Yeah, for this set of variables, we might only need to measure mouth diameter, and it looks like that height doesn't vary across sites.
Yeah, and if we were focusing on something else, maybe besides plant height, we might see that sites do matter, right? Here We're just looking at plant height.
We could have no outcome variable, in which case all we're doing is looking at the patterns of the shared variation between a set. And this is PCA, principal components analysis. And so in this case, we're going to leave site out of it, and we're going to look at how height, mouth diameter, tube diameter, and keel diameter are all related to each other.
Yeah, how do they covary, which ones are more related than others, etc. And we can visualize that, if you recall from Quant Methods 1, we can use a plot like this to take a look at what's going on with those variables.
So we see that, for example, tube diameter and keel diameter seem to change in the same direction, whereas height and mouth diameter change in the same direction. And the magnitudes of those look like they're all about equal. Again, we'll get into lots more details later, but this is the kind of question that we would then, we could actually then do something with these principal components if we wanted to.
We could have multiple outcome variables. So this, might look very similar, but note that we've got multiple variables on the y side of the equation here, and those are all being predicted by site. And when we have the case of multiple variables on the y side being predicted by a categorical variable, remember site is a categorical variable, that is called MANOVA.
Multiple ANOVA.
Yes. Mm-hmm.
And so, we're going to basically ask the question, jointly, how are all of these variables predicted by site?
Yep.
And it turns out pretty well. So there's significant site-to-site difference in the combined set of variables, right? So it's not just one of them, it's all of them together.
Yeah, we already saw that it seems like plant height isn't impacted, so you might have some predictions about how these other things might be impacted by different sites.
And we just saw in the PCA that they actually do covary with each other, so again, it's all kind of coming together there.
Okay, we can also have a categorical outcome variable with multiple predictors. So this is very similar to what you just saw with the MANOVA. I'll just toggle back there, except that now site is on the y side and our multiple predictors are on the x side. And so it's really just sort of flipping that question, and often these are done hand in hand. People will do both and then interpret them and try to learn from them together. One approach to this is logistic or multinomial regression. So we fit a multinomial regression here because there's more than two sites. When you have just two, that's sort of a special case, and it's called logistic regression, which you also have experience with. Mm-hmm. And then we can look at how each of those variables are impacting our ability to predict which site it's in.
Yeah, and so here in our output, we've got, these are the different sites, and then these are the different variables predicting it. And so a lot of this table should look familiar. You've got estimates and standard errors and p-values and confidence intervals, right? So it's the kind of stuff that you've seen before.
We can do exactly the same thing in another way, and that's called discriminant function analysis. The two of those are really very closely tied. They are essentially doing the same thing. They give you slightly different output. It's a lot of sort of what we've already told you many times over, that many of these models are really kind of in the same family, right? Mm-hmm. And so here, again, our equation is the same. It's just a different approach to this question of predicting site from multiple predictors. And one thing that we can do after a discriminant function analysis is we can actually do some cross-validation to see how well the model actually does at predicting site. And so this is essentially holding out some data at a time and then seeing, okay, how well does our fitted model then predict site? How well can we predict where you are based on your values in these different measurements? And we can see here that it actually only gets about half of them right. So just about, this is a percentage down here, so about 50%. So not so great, actually.
Right. So, but this actually, yeah. So it might tell you something about, right. So all these DG ones are probably mostly good, but some of them are categorized as HD or LEH, and the most are TGH, right? right? Most of the LEH are correct, but then some are thought to be one versus the other, right? So this is kind of how you interpret this table, and we'll again come back to this in future lectures to talk about more about DFA. And so DFA and multinomial or logistic regression are very similar to each other in that they are doing similar things to answer kind of related questions, but not quite exactly the same.
Yeah.
Just a couple of references where our data came from. And then again, Tabachnik and Fidel is a good resource for multivariate statistics.